quick preview of what the course was about. Before I do that, however, one of the things that I have to finish is what we left in the last class. And this part is unfortunately a bit dense in the sense that it requires, there's a lot more biochemistry going on in terms of how much you will be able to understand and how much I will be able to do justice. So for this part, if you don't understand the full biochemistry of it, it is okay. The reason I want to still discuss it is so that you're aware that something like that exists, right? So it's that way, it's still part of the exam, but of course I'm not going to ask questions like which reagent is used where and so on. It is a bit technical and it's a bit dense when it comes to the biochemistry. The goal is not to understand the biochemistry part for this lecture, but to understand very quickly how to profile different marks. Right, so I'm sure you're here, so one of those things which will really important is for you to know what logistics of the class. So the exam is on 18th. This is in class. There's no, it's open, it's closed book. There's no notes that you can refer to. It's going to be a very simple quiz. It's 12.5% of your overall thing, so consider it more like a quiz. It will require, I will upload the slides. So you will require you need to know the material. The material does encompass everything because I think it's very hard to distinguish, at least for the sequencing part, what happened before and after the midsem. As I said, I will not cover a lot more mathematical questions in the last part. It will be very thought related questions where you do thought experiments. If the kind of one which you have in your assignment, for example, that's a thought question, right? There is no right answer there. I just want to see and just think with you that how do you think overall. Okay. You have we gave 6 assignments. We had, the initial number was 8. We cut it down to 6. Only 5 out of these 6 will be evaluated. The midsem was 22.5%, and sem is 12.5%, so it's more like a quiz because the surprise quiz itself is 10%. The course project is of 25%, right? So that is something that you should be focusing on more. The deadline is Friday 9 a.m. and usually the deadlines are at 9, but the reason it is 9 a.m. is because for me, the grading deadline is 1st December, and I need to have some time to actually finish things. So that's why it's in the morning of Friday. Okay. Any questions about this part? Okay. So if not, we can begin. And this is the last class. So this is the first slide that I showed you when you actually came in, and this is also the last. The first slide of the last lecture that I'm going to see, and hopefully in the process that you took this course, you have learned something new, have discovered something new, and the goal for today is to go back and revisit some of these ideas, trying to connect the dots. But I feel that, I'm sure some of you will feel that you've actually just gotten started. The field is moving extremely fast. There are a lot more things that are happening which we unfortunately did not have time to cover. But the principles that we learn in the class will be something which hopefully will stay with you, and these are general principles which will apply in whatever field you choose to work, or whatever sub area you decide to work in the area of genomics. So the goal was to teach you or give you a flavor of science despite being in an engineering school, because science works slightly differently than engineering, and engineering approach is more like, this is the problem, you go and fix it. Science takes a slightly different approach and goes a bit meta, trying to ask how and why did that problem occur in the first place. Can we come up with first principles based application explanation to whatever we are observing? Why does it act like that? Right? So every part or every few lectures we have seen that flavor of something happens, but why does it happen, and can we study something about it? That's what we have been discussing overall. So hopefully you have this fundamental analytical framework that you can answer your own questions you will not need me anymore, right? You have gone through your enough training to actually be somewhat independent, where you can actually come up with your own analysis, and you'll be able to think about different questions that you can answer yourself. Right? So the project is one such example of the things that you'll be able to do on your own. You don't actually need me to be able to do that. Neither do you need anyone else to teach you, because hopefully you have all the you have already gotten a hold of the basic principles that are required, and you always can build upon it, right? So you don't, now you have learned, if you learn 2 + 2 is 4, you don't need to go back and learn about basic mathematics anymore. So that's what the course is designed to be. Of course, how much you take it will be will depend on different people, and it's also a function of how I taught some particular lectures. But the lecture slides, if you go back, I think there is enough content for you to actually go deeper if you really wanted to go down rabbit holes trying to figure out how something came about to be, where is the field going, and how things are progressing overall. Yeah, so this I've already talked about. Science and engineering is slightly different, not fundamentally different. There is a top-bottom versus bottom-down approach, and the reason I'm highlighting this is because my first 2 years of grad school I suffered a lot because I went through an engineering mindset. There's a given problem, you try to fix it, and you're happy with it, but we're not trained actually to ask, why does it happen? Why did it begin at the first place? And is there something that we can actually fundamentally do which will change the nature of that particular problem? Right? That's what the difference between science and engineering to me is. Everybody will have a different flavor of what they understand about engineering. I'm not against engineering, and I'm also an engineer, but I think there is one fundamental aspect that we should borrow from science is actually how to think about fundamental problems. If something happens, not take it at its face value, and really go deep and think about why does it happen. Okay, so we'll quickly go back and finish the epigenomics lecture. So there's no review, of course, for epigenomics that we started in the last class. It is a dense lecture, and I realized it only when I actually started to talk about it last time, that there are a lot of concepts which I will not be able to do justice to. So that will unfortunately happen today as well. The goal, as I mentioned, is to get you a flavor of, if you wanted to tap into the cell and measure before gene expression what changes are happening, that technology does exist, right? There is lot more things that are changing. There are a lot more reagents that you require. There are a lot more concepts that you need to master. But the idea again here is to take this approach where we can understand that if you wanted to do it, how would you go about it? Okay, so we started talking about these different marks, the acetylation marks and the methylation marks, and that itself is very confusing. Right? There are histones, there is DNA, the DNA is wrapped around histones. Histones are made up of amino acids or proteins. We have a tail. The tail is going to get attacked, or is susceptible to being deposited with different chemical marks, some being methylation and acetylation being the two more common ones. And because of this, other proteins can either come in or cannot come in for the RNA to express. Right? That's the fundamental principle on which the field of epigenomics works. You deposit a chemical mark, and because of that chemical mark, some other changes happen in the vicinity. So for example, to transcribe the gene, this is the transcription start site, which is called TSS. TSS needs to be bound by Pol2, and this Pol2 can actually benefit if there is other proteins which are in its 3D vicinity, which is what generally is marked by these enhancer regions. For this to happen, this particular nucleosome which is sitting in its vicinity needs to have certain chemical marks deposited, so that it ensures that this region that you're seeing here right now will be open, because that should be a region which does not have nucleosomes. If you have nucleosomes sitting in a particular region, as you can see from here, it's very hard for any transcription factor to come and bind in this region. Whereas if it is nucleosome free, for example, regions like this, transcription factors can come and bind. And why do we require transcription factors to come and bind? Because they're trying to carry out this synergistic reaction where while Pol2 is sufficient to start producing certain copies of RNA, if you require more copies of RNA to be present or to be expressed, you need an enhancer mechanism, right? Just like if you've ever seen those trains, the train can run with one engine as well, but if the train's load is extremely heavy, you will have an engine at the back as well trying to push it. These enhancers are exactly that if you want to enhance the speed, if you want to reduce the overall load of everything happening at one level, and if you want to enhance the overall final output, you enhance the expression by giving it space. This space will be occupied by transcription factors, which will then allow you to actually have more copies of RNA being produced. These regions are usually marked by different histones. Histone 3 chain, again, this is one of those histones. This is one of those histone proteins which makes up your entire nucleosome. If one of those tails, which is H3, is marked with an acetylation mark, it will usually mark that this particular region is active, which means that in its vicinity, transcription factors can come and bind. Right? So there is still a nucleosome overall, but overall this mark that you will see, right, there will be a full chain of marks in terms of how it will get deposited. So all these different nucleosomes in this vicinity, for example, if they all have an acetylation mark deposited, in its vicinity you will have a lot more transcription factor binding to happen, and that marks an active region. On the other hand, if you have the same in the same histone on the 27th lysine, or the 27th position which has a lysine, if you have a methyl mark deposited, then that marks the repression. So H3, K27, M3 will lead to suppressing the amount of protein, the amount of RNA that you're going to synthesize. So there's a bunch of things going on here, but what you want to take away from here is overall what happens, that if you have an inactive promoter, which means that it has these particular Me3 marks deposited, which causes the transcription factor to not come in. If it cannot come in, the amount of RNA, it's not going to happen that you're not going to synthesize any amount of RNA, but the basal amount of RNA is all you will get. You will not be able to synthesize more number of copies of the mRNA. This is the promoter site. A similar thing can exist at the enhancer site. So if this interaction that happens overall, the transcription factors can also come and bind in this region. This is the promoter region. They can also come and bind in the enhancer region. So if those marks get deposited in the enhancer region, it becomes an inactive enhancer. If these marks get deposited in the promoter region, it becomes an inactive promoter. But the general concept on which they are acting is similar-ish. Overall, methylation marks, density of nucleosomes cannot be, density of nucleosomes is high, chromatin becomes compact, TF cannot come and bind. And that's the general principle connecting all of these. If you have the acetylation mark, on the other hand, it makes it accessible to other proteins. Things will get transcribed, whether it's in the promoter region or it's in the enhancer region. We will see the difference in just a bit. Any questions about this slide? Yeah.
So, we know the sequences of the promoter region. Do we know the enhancer sequence?
Yeah, that's a good question, right? So I told you something like the enhancer exists. We know the promoter because we know where the transcription usually begins from. So I can ask you this question: that how do you figure out where did the transcription begin from, right? So it's a mapping problem that if I take this data and if I map it, if I have the annotation of the genes, then the transcription start site is the one which will essentially mark. So if this is the transcription start site, I will see reads in this region, but I will not see reads, RNA sequencing reads coming from this region. Right? That marks the transcription start site. But how do I mark out? So I can, if I know the transcription start site, we can take a guess that the promoter region is somewhere in its vicinity, and we still actually don't know what is the definition of a promoter. Some people say that, or some research studies say that you have 1,000 base pairs or 2,000 base pairs in the vicinity of transcription start site where transcription factors can bind, so that is the promoter region. We take that as at the face value that 2,000 bases near the start site is what marks your promoter. The enhancer is a much difficult problem, because the enhancer, for something to be able to be called as an enhancer, you need a direct proof that this binding is indeed happening, right? This structure of bending over is really, really happening. And there are assays to do that now. These are called high-C assays, which I have not again talked about. The idea behind high-C assay here is that you can, if this contact is happening, you can treat it with certain chemicals so that that contact becomes intact. And now you can cut this here because you can break this DNA here and you can sequence these fragments. You will essentially end up sequencing only the promoter region and the enhancer region if they were in contact. So the ideal technology, which I've not discussed here, to calculate or to figure out whether the enhancer and the promoter are talking is to come up with something so that these two get glued together if they are indeed talking to each other, and then sequencing only those parts and deleting everything else. So that is done in this assay called high-C. That's how we know the actual definition of promoters or enhancers. However, again, you might, if you don't have access to the technology like that, what you know is if the enhancer is, if you were to profile this region and you were able to see that there are certain regions in the genome which are open, which means that they have open chromatin, which means there are technologies to do that. I'm going to very quickly talk about it. But if you can somehow measure the nucleosome density in these regions, a high density region and a high density region in its vicinity, and that vicinity is like 100 kilobases apart, or maybe even 1 megabase apart, that becomes a proxy for me to call something as an enhancer. So if I take this particular thing, I of course know where the promoters are because I know where the transcription sites are. I can calculate with different technologies that I'm going to talk about in just a bit. I can calculate whether these regions are part of the genome which is accessible, or are these part of regions which are actually closed. So is it part of open chromatin or closed chromatin? That technology exists. If I can do that, what I will notice is the moment this is open, I see more expression here. But if it is closed, right, I don't know the enhancer, right? But I know in its vicinity, if I go 100 megabases apart or 1 megabase apart, where does the openness of this tell you how much amount of expression you're getting? So if you can correlate that expression, that becomes a proxy for you to identify what could be a likely enhancer for a particular gene. Okay. So this will become clear once I talk about this technology, which is called ATAC-seq. Okay, so usually if you have the genes which are being expressed, they will be part of open chromatin. Again, this is similar plot but repeated. You saw this version in the 3D in the video that I had shown. There is something called the insulator, which is insulating the active part of the gene from the repressed part. What you notice in the repressed part is there are these methylation marks, H3K27Me3 deposited or H3K9Me3 deposited. 9 and 27 with a methylation mark means repressive, and K4 which is on the top with any methylation mark or acetylation mark, H3K27 acetylation. I'll give you a mnemonic to remember this. It's not that you have to really remember this, but this will keep surfacing again and again. It's highly confusing because, you know, if you think about it, why 27 and why 4, and I don't think anybody knows. 4 and 27, they happen to be very special numbers. So if 4 and 27 methylation and acetylation respectively, you see that is part of your active region. Everything here that you're seeing is not in 2D, it's happening in 3-dimensional world. So the genome has this capability to actually separate out the expressed part from the non-expressed part. And this is technically what happens in your tissues. The liver expresses certain proteins or certain RNA, the brain does not. That's because in the brain, the DNA has not changed, but it is put into a different compartment. Those genes, which are specific to liver, are repressed because they don't have to be expressed in the brain, because they're now part of the other region which is fully repressed, which does not require transcription. So this is how, in a sense, what happens in different tissues. This is one of the mechanisms by which tissues are able to express RNA which is specific of their type, right? There are a lot of genes which you will only find in the liver, expressed in the liver, but not in brain or kidney or lung or anywhere else. And this is one of those mechanisms by which it is able to do that.
But why do you put the effort to translate that from DNA to RNA that is not required to be expressed?
No, so that's what is happening, right? At this point, you're not expressing RNA. This is the string here is DNA. The DNA has been compartmentalized such that the genes which would have expressed the brain specific RNA, for example, these genes are nothing, these genes are expressed everywhere. I mean, these genes are present everywhere because they're part of the DNA. But when they are put in a compartment which is not expressed, for example, in liver. Right? So if I'm looking at the brain, then assume that these are genes which are only expressed in the liver, and they are part of the system which will not be expressed, right, or very minimally be expressed. Similar things, which again happens in cell types as well. This is an extreme example of two different tissues, but we saw that when we looked at C1QB or C1 or ApoE, which C1QB, which marks the microglia of the brain, which was expressed only in microglia. The reason that could happen was there are multiple mechanisms by which it could happen, but this is one of such mechanisms that the epigenetic rewiring that's happening in those cells is allowing the same gene, it's expressed in the same system. Now the tissue is also the same brain, but it does not express because it happens to be sitting in a region which is being repressed because there are other epigenomic marks happens to repress. Okay. Okay, so this part you will see again and again. I will not emphasize it. CTCF is called an insulator. This insulates the active part of the genome from the repressive part. This is that loop exclusion that happens. So you can think of the CTCF as a ring. Among the ring, you have the DNA being coiled up, which allows it to contact one another. But the other part of the ring, which is what you see, for example, here, is repressive because the coiling is happening such that the proteins, because of the deposition of these different acetylation and methylation marks, you cannot, just the methylation marks, you cannot have these kind of contacts which are required for the gene to be expressed in this part of the gene. Okay. Does that make sense? The insulator part is trying to insulate the expressed part from the unexpressed part, right? So you're creating a 3D environment where the crosstalk cannot happen between what is being expressed and what is not being expressed. So it's insulating the active part where the contact is happening from the part where there is no contact happening between the enhancer and the polymer. No, CTCF is a protein, but it is called CTCF because if you look at its motif, no, actually CTCF does not have that example, but there are motifs which there are genes which are named after the motif that they bind to. So this protein called GATA, G-A-T-A, that binds to a DNA sequence which is also rich in G-A-T-A. That's the motif that it binds to.
So wherever CTCF sits and sits, that part is beyond the insulator.
Yeah, so this entire part is the insulator. So this is kind of, you can think of this as bringing up walls inside your DNA, right? So this is literally a wall beyond which you cannot actually overhear what's going on in the other part of the, right? That's where the insulation is coming from. Okay. So it's a protein. It binds to certain DNA sequences. You can actually also profile. We'll see that technology. How do you actually figure out where does it bind? Right. It does bind somewhere, and it has its own preferences. We can figure it out where it happens, right? So this diagram is like just a repeat and repeat of what you have seen previously, but this is a simplified version of things, right? This is your exon, intron, exon. This is the gene transcription start site. If this region happens to have K9 methylation marks, you will have or K27 trimethylation. This is called trimethylation. Then this part is repressed. If you have, on the other hand, K27 acetylation in this particular part, for example, this is K27 or this is K79, K36. You don't worry about all those. But if there is any sort of K4 methylation marks, usually that will be part of the active region. We will see again a slightly more simplified version of it. But K4 methylation usually is associated with active regions. K27 acetylation also is usually associated with active regions. Okay, so why so many marks exist, right? And this is one clear depiction of what really happens. And you can see that it is complicated and there's no way you will be able to recall and remember everything, and neither are you expected, neither am I expecting you to. But overall what happens is the reason these mechanisms are devised is to give you control of how much fine tuning control can you do with the expression of each gene. So at the promoter and the enhancer are the ones which are more important. Like mostly you will look at marks which are not sitting in your protein coding regions. These marks can also sit in the protein coding regions. It's extremely complicated. We still don't understand how to interpret it because these change very dynamically. By the way, we are still focusing on histone marks. You know, this is just one layer of control that happens. You also have DNA methylation, DNA ME, which also will play a role. But overall, if you have DNA methylation on and if your H3K9 is methylated at the enhancer, then it becomes an inactive enhancer, which means that your gene expression is overall going to be suppressed. On the other hand, on the other extreme of the enhancers, if you have your K4ME methylation mark deposited, you will get or H3, the K27 being acetylated at the enhancer level, then that is mark of an active enhancer. Similar approach here as well. Usually K4 methylation is indeed associated with, that's why I was saying usually, it's associated with active mark because K4ME2, K4ME3, if you happen to see in the active region, is also representative of that particular gene is highly active. On the other extreme is again your DNA methylation and K9ME3 and K27ME3. Okay, so you can see that now with K27ME3, you will start to see suppression. So there is no exact code that 27 is the universal number or 4 or 9 is the universal activator. That unfortunately does not exist. There is some amount of requirement that you have to do, sort of figuring out to do here what is.
Sir, what is recommended by inside the
Oh, so sorry, I will explain, right? So if you have a DNA methylation, so what this means is if these levels are high in a particular region, then that is part of an inactive enhancer. Right? Yeah.
No, I'm asking about post K27ME3. It's given in the middle.
Oh, so poise, okay, sorry, yeah, thank you for asking that question. So poise is somewhere in between, which means that it is neither fully suppressed, neither fully active. It's poised, so it's about to be active. If you do that one particular change, for example, if it goes from K4ME3 to K4ME2, you remove one of those methyls, for example, or two of those methyls, it becomes active. Right? So poised, again, this is an active area of research, what are called poised enhancers. So this is, again, if you think about it, why would somebody want to be poised? Any ideas why should that intermediate level exist? Yeah, it's like an emergency situation, right? Something goes on, you know, or you know. Yeah, it will be activated, right? So this happens a lot, by the way, in during developmental time course, because at the end of the day, you're all developing from one cell, right? So as you develop from one cell and as it starts to differentiate, these are the subtle changes that are happening very transiently, so that you can again make fine-tuned controlled expression changes starting from one level of. Yeah, it's not, so you can sit around, right? The poised is like, you know, it's ready, it's kind of doing its job, but it's not either on the other extreme side. So either is it suppressed, so neither is it making very fewer copies of RNA. It's there, it's doing its job, but it is about, if required, it can actually be leveled up. Is it getting hot? It is getting hot. I think the AC was cut off. This is fan. Anyone sees AC control? It's there. One second. Another message. There are no fans also, right? So, because I can feel it here, but I don't know if you can feel it. It's actually pretty hot. Sorry, I'll just check that. It's not here. Okay, hopefully it'll come and switch it on. Sorry about this.
What if you talk about the risk scores? Can that be correlated with the voice?
Oh, the risk scores?
Yeah.
You mean the polygenic risk?
Yeah, the voice.
Oh, yeah, sure. Yeah, so there is an area of research which actually tries to sort of look into it together, like trying to think about if you do get a, if you try to get the mutations, for example, what happens to the status of the enhancer? And sometimes what you see, for example, particularly in the area of vaccine development, right? There is, for example, papers out there particularly studying COVID vaccine. COVID, some people respond, some people don't respond, right? Or it gets very severe, for example, even after they're being vaccinated, right? So, for example, I took vaccination twice, but when I actually got COVID, it was extremely severe. So what happened, right? Why is that response so weird, different in different individuals? And one of the, I think, if I remember it right, one of the findings was that in certain regions, one of the enhancers which should have elicited the response was like based on the genetic profile was not in the active state but in the poised state, right? So the response was not as sudden in terms of what it should have been, or the other way around. Like I don't remember the direction overall, but that's one area where people are looking into what happens given a genetic mutation or the epigenetic profile. How does it inform what happens during a particular reaction?
Just a follow-up. How dynamic will be the process of turning point?
It's very, very dynamic. It can be very, very dynamic, right? So, particularly in the case of, it really depends on the system that you're talking about. In the blood system, it can be very, very dynamic. It changes very, very quickly when required, particularly in the immune cases, right? So that's why the study was particularly done in the vaccine case. I think it's also seen a lot in the developmental time course, right? So if you do a developmental time course, you'll see that a lot of enhancers which are now at some point going to make a decision to make two different cell types. That happens because there are enhancers which can quickly, so some diversification happens early on, right? So brain is different from limb, for example. But limb, if you start to make fingers, that diversification is happening later on. So the first diversification is quick, so those enhancers will be at the poised stage, and the later ones can be, it can be a switch on or an off. Right? So developmental biology, which we did not touch in the class at all, is one of those areas which single cell, for example, uses a lot because it's very, very dynamic. So there are a lot more interesting studies that you can do in such a system. Okay.
So,
Yeah.
is there a reason why we observe these modifications of this 2, 3
Not on other ones. Yeah, it's a good question. We should study I don't know the right answer, but I think it's probably has to do with the context, because each tail and the the monomer is different sequence structure. So maybe the way it is structured allows this particular H3 histone to be accessible to these, because at the end of the day, some enzymes are the ones which are going to bring out these marks, right, and deposit it. But if the other chains are, let's say, coiled, then they will not be susceptible to come and get deposited, right? So I don't know the right mechanism. I'm just trying to think it out aloud. That could be one way. That how they're organized again in 3D. What is their structure? That can, if it is a very close structure, then no marks can be deposited. If it is somewhat, like a tail is literally like a tail, then it's open, then we can come and deposit. But I don't know the right answer.
Do we know the physical structure of
Yeah, we know. I think we know, right? Because now there is all sorts of fancy stuff, including collab fold. You can fold anything that you want, so it's not hard to actually check. I've never checked the structure of these histone proteins, but it's a very well characterized. I think it's a very well characterized structure, but I'll double check. Okay. Any other questions? Okay. So that all the slides were a bit complicated, but this is one way to sort of think about it and remember what is happening. So on the top you see chromatin accessibility. This accessibility, if you see high, these are called tracks. That represents that wherever you're seeing like a peak, that means that that particular part of the genome is accessible to chromatin. So it's high means it's accessible to chromatin. So that there are different regions there: promoter, enhancer, which is poised or primed, insulator. So nothing, you'll see particular things at the insulator side. Then comes your active enhancer, inactive promoter, inactive poised promoter. The rest of the stuff does not matter. Active promoter matters, and the enhancer active matters. Right. So look at the marks which determine. Right. So if you are Me1, HCK4 Me1 or HCK27 acetylation, they are both present at active enhancers. However, if you focus on this part, which is way above, which is slightly above the towards the other insulated region, then those are both active. So one of them is active at that particular region, whereas STK4 Me1 is active only at the enhancers. Right. So there is a permutation combination of things that can happen based on which particular part of the genome you are at. If you focus on the insulator part, then both K4 Me1 and 27 acetylation are accessible. This track shows you where does CTCF bind. Right? So CTCF is bound at the insulator site. There is no other region where it is binding. So this becomes the insulated part, for example. This part is, it's very far away actually. That's what this shows. That's why this stuff that you're seeing is actually it's active in this case. But if your promoter is inactive, then in this case you're not seeing any of these marks. So lack of a mark essentially shows that that mark is not present. If, however, your HCK4 Me3 is present, but HCK27 acetylation is not present, that's a poised promoter. Right? So you don't have to remember all these. Adjusting one. Vijay ji. विजय जी बहुत गर्मी है बहुत ज़्यादा गर्मी है Sorry, it's also getting recorded. Okay, so if you focus on, for example, in this particular part, right, K4 Me3 and K27 acetylation are both active. That marks your active promoter. The enhancer, on the other hand, is K4 Me1 positive and K27 acetylation positive. Right? So there is a combination of what happens and what marks what. I will not expect you to remember this because it's hard, and you don't have to remember it because you can always look up this track and you will know what happens where. Right? But hopefully you will be able to interpret what is going on. Right? So if, for example, if this structure is given to you and you're asked this question that, if you can assign the right labels to each of these things, that kind of stuff, hopefully you should be able to do. If the rules are overall, noted. So what is that rule that you want to remember? That rule that you want to remember is, it's kind of summarized. This is again still a slightly complicated plot to remember. But you focus only on the H3s and H4s to begin with. Here, there is no accessibility of this transcription factor. Why do we require transcription factor? Because that's what is going to help us boost the expression overall. If your DNA is methylated, so if this is yellow, that means the methyl mark has been deposited on the DNA. In that possibility also, you're going to be, those TFs will not be accessible, they will not come and they will not be able to bind your DNA sequences. So you will not have gene expression. The other reason this is the repressed part where TFs cannot come and bind is because your H3K9 has one methyl mark deposited, right? So this is that mark. So on the left side is why transcription doesn't happen. On the right side is where the transcription does happen because TFs can come and attack. Now this is complicated because this can happen at multiple different locations, which we have not even talked about, which is H4 acetylation overall. But there is the one thing that you can remember is H3K9 methylation and H3K27 acetylation. That is going to be often associated with overall active region. So there is permutation combinations of it. You don't have to remember it, but just remember that if there are certain marks which can allow you to help you to control or to fine tune the expression of your gene overall. Right. So the only thing you should remember hopefully is when K4 is methylated, your genes are activated, but thiazole 9 and 27 combine to keep genes inactive. So K4 is methylated, your gene is activated. If you have if you have a K9 or a K27, then your genes are going to be inactivated, right? This is what is represented, for example, in this figure here or this figure here. Okay, this is only about the methylation part. The acetylation is hopefully very, very clear. Though that's the only place where you see. This is the only place where you see activation. Acetylation. Okay. Okay. Any other question about this? Okay. So that was what was being deposited at the histone level. You can also have deposition of methylation marks at the DNA level. So this is your enhancer, promoter, gene, and your transposon, which you don't have to worry about right now. Overall, at the promoter level, if the gene has to be expressed, then your methylation level needs to be higher, right? So this is CpG, and the red one. The red one is CpG, and CH, where H can mean anything, right? So CG are regions which are often found a lot in the DNA. These are called CpG islands, for example. You've seen the example in one of your assignments, and I think your mid-sem as well. Now these regions are also ones which are often bound by a few transcription factors. There are regions which are CG rich, which are preferred by transcription factors. Now if these regions are by default methylated, then you will not have expression. But if these regions are not methylated, then proteins, transcription factors can come and bind and allow you to express the gene. Right? So this overall shows what is the overall levels of methylation, which is between 0 and 1. So at promoter, you don't see any methylation. What you will also notice is what happens at CpG is also usually followed everywhere else except in these regions, which is because those are CpG rich. But overall, at the promoter site, you will not have, if you want to express your gene, you will not have any methylation. Okay, so we have seen that there is some biochemistry going on, which allows us to measure what happens overall. In the next 15 or 20 minutes, I will very quickly walk you through what technologies exist, right? So to profile, if you wanted to profile this. So just pay attention to what is the central idea. Don't worry about like what reagents are being used or what one particular thing is doing. I will explain this as we go along. This is slightly dense, but we can do it in the next 20 minutes. First thing that you want to measure is DNA methylation. The idea behind measuring DNA methylation is very, very simple. You want to measure, usually the methylation will be deposited at CpG sites. So this sites which are C followed by a G. If you want to measure this, you do what's called a bisulfite conversion. The bisulfite will convert the unmethylated C's to T's. So that's what you're seeing there. The first C is methylated, nothing happens. But in the next, in the unmethylated C, that gets converted to a U. So I should use that gets converted to a U, which when you sequence will become a T, right? So when you start to sequence these reads, if this particular site is actually methylated, then you will start to see a T coming from here. Whereas in your reference sequence, you know that is a C because somebody has already created a reference. So we know what that is a C. So if we can come up with an aligner which allows this mismatch, C to T mismatch is allowed in the aligner, you can align all the reads and go back and see where did I see a C to T mismatch. That regions are ones which are methylated. Everything else is CpG sites are not Does that idea make sense? It's very, very simple idea and works very nicely. This is what is used, for example, in what's called whole genome bisulfite sequencing, or RRBS, reduced bisulfite sequencing, which we will not, of course, will not be covering in the class, but that's the central idea that basically connects both of them. You cannot, in an ideal world, you would want a technology which can actually detect methylation, right? You should be able to measure methylation directly. We cannot do that. So we come up with a proxy way to measure it by converting the unmethylated sites to a different base, and then using that as a leverage to figure out how many sites are methylated versus not methylated. Okay, this is what is done in all DNA methylation technologies, except nanopore, which can actually detect methylation signals. So if you recall nanopore sequencing, it works on the current idea. Now, if the current will be different from a methylated C to a non-methylated C, you can actually see it changing slightly differently in the current. So you can measure it directly. And otherwise, when you're doing sequencing, you can only sequence things. Then you have to come up with a proxy way to measure the methylation mark, which is done here using a conversion step. Okay, very quickly, and if somebody sees this, they will think, what am I doing? Because this is like, if you're in class 1 now, this is class 10 material, to actually walk you through all the steps that are happening. But again, as I said, this is just something that you should be aware of. So we want to measure where proteins are binding in the DNA. What are these proteins? Proteins are transcription factors. What are these factors? These are factors which help you enhance the expression that you will see from a gene. These are called TFs, or transcription factors, which look like these green material here. Why do I want to measure it? I want to measure it because I want to figure out that whether a gene is particularly is being expressed or not, if its promoter or if its enhancer is being bound by proteins of interest or not. So certain genes can be on and off despite all of them being fully open. It can happen that if I have one cell type, this is my cell type and this is my cell type 2. It can happen very well that even though both of these are DNA, is open, the transcription factor which needs to come and bind here could only be expressed in this certain cell type. If I do a study, I want to figure out that this transcription factor of interest, does it really bind to this DNA? Because if it does bind, that will help me explain that why is that particular gene being expressed. So this kind of study is done to figure out that what regulates what, right? So this is called TF. It's helping me regulate the expression of this gene. So I want to figure out where all does it help, if you want to think of gene expression as a network, is it regulating gene 1? Is it regulating gene 2? I want to figure out which master regulator. This is a master regulator in the sense that it can actually help regulate multiple of these genes. Where does it bind? Right? These are proteins which you already know about. You want to figure out where do they bind in the DNA sequence. This is done using this technology which is called ChIP-seq, chromatin immunoprecipitation. The idea behind this is nothing, very, very simple, that if the TF is bound, if it's binding to a particular DNA sequence, you treat it with certain reagents so that it will become like this. It will become tightly bound to this particular DNA sequence. It's not going to move apart. Once it does, it's going to capture so this is actually bound. That means there is definitely sequence here which you cannot see. But if you end up digesting everything else, right? If you digest things which are not being bound by a particular protein, then you can sequence this particular part, this short reads that you get from this particular part, and that will tell you that in that particular region is where binding is happening. So very simply, what it is done, this step is called cross-linking, where you cross-link the protein to the DNA so that it does not move out. Once that particular process happens, you put in certain enzymes. These are called endonucleases. These endonucleases are nothing but they're going to chew off fragments which are actually open-ended. Right? So this is endonuclease. They will chew off fragments in the direction where there is a closed end. So it starts from here, it stops here, it starts from here, it stops here. What remains is these DNA sequences, which you can actually now put again on your sequencer. The first, however, you have to make it a bit more specific, right? When you do this, you will be able to target all kind of proteins. Right? There are like 1600, 2000 level of transcription factors. I don't want to study all of them at once because that will not tell me anything about one particular protein. What is it doing? So to only sequence or to only select certain proteins, you use this idea which is of immunoprecipitation. So you add an antibody. That antibody is something which is specific to that protein. So it will only attach to that protein. So you're adding again one more reagent which can only identify the transcription factor which you are interested in. So it will go and sit there, and next you let it settle down. So that's where the precipitation happens. This will become heavier, will settle down, and you then can come back and sequence these fragments. And that tells you where exactly where was the original protein of interest found. If you don't do this step, right? If you don't do this step, then you will get all sorts of proteins which are bound everywhere, right? So there could be multiple proteins which are bound here, but I'm not interested in all those. I'm specifically interested in this one because I know I want to study the properties of what does this protein do in my system of interest. Yeah, so here at this step, it is taking in information about this antibody is designed specifically against this transcription factor, right? So this is very similar to what happens, for example, in the case of vaccination. Vaccination is designed for a particular kind of strain of immune response where we know about that. This is the kind of foreign body that if the immune system sees next time, it will be able to design and attack it directly, right? So this receptor that you have is specifically going to pick out that particular transcription factor. It will only attach to that particular transcription factor. So we know we have to design this experiment based on which transcription factor we are interested in. So there are like 1600 or 2000 proteins which are called transcription factors, which come and bind to the DNA sequence. Okay? We know their properties, so we know what antibody acts against those proteins. So this is the antigen, that is the antibody. And that antibody is what you are designing. So you take it one from the market and say that, okay, this antibody I know acts against this particular antigen, which is my TF of interest. If I can attach this to, if I add it to my mixture, this will get attached. The entire mixture will settle down. I can then figure out where all was this protein of interest bound. Okay. You can do a similar reaction with the proteins of interest. So this is going to tell you where does the transcription factor binding happen. Now again, the reason we are doing this experiment is there is no technology in the world which can tell where is this transcription factor being bound in the DNA. So we are using again the proxy that if I can sequence and somehow selectively target this, I will know where it is bound. But in an ideal world, you should be able to put all of this through a technology which will tell that this TF is transcription factor is bound at this location, the second transcription factor is bound at some other location, and so on. But that does not exist, so we are trying to leverage sequencing to be able to do those experiments, right? So it's extremely innovative in how these experiments are designed. To leverage one system, this is the engineering problem of some sort, where you have a solution in hand, you're trying to see how you can engineer your system to work towards that particular goal. The second technology there is it's basically the same technology, ChIP-seq, but now you want to understand when you had these histone tails. This is H3. I want to understand, or I want to figure out whether is this K27 acetylated. Is it 27th lysine acetylated or not? Again, if I have access to a technology, I will say that look, let me pass through that technology, and I can read the signal, and it will tell me whether the 27th lysine is acetylated or not. But I cannot do that because that technology does not exist. So what you do is you use a very similar idea, which is again, if this histone is acetylated, to design an antibody which can attach to this particular kind of histones, right? So these antibodies are easier to manufacture to some extent because all they're looking for is they're looking for the structure of the protein. They're looking whether that is acetylated, and if it is acetylated, it will go and attach. This is what is shown, for example, in this reaction. Once that happens, you can again do the same thing that you were doing in this case, right? So the only difference is how you design the antibodies. You can design it to target transcription factor. You can design it to target marks which are deposited on histones. Right? Does that make sense? Transcription factor is also a protein. Histone is also a protein. You can design specific antibodies that can also look for these proteins which has that particular mark. Right? So that's the engineering part of this problem. That's a biochemistry problem which we'll not talk about. Okay. Okay. So there is a different way to do this, and this is slightly complicated. You can again use a very similar idea to do this, but the problem with the technology on the left is it requires number of cells. So if you have a very small tissue, for example, and if you start to do ChIP-seq on it, you will lose a lot of cells. So you'll have to start with millions of cells to actually get any amount of readout. One way this is solved is by this technology which is called cut and run. Cut and run, what it tries to do is it uses the similar idea that you still have to design an antibody, but along with that antibody, you have a special kind of. So this is the first step where you will be incubating it with a particular antibody. The second step here is you treat it with a MNase. This MNase is like an enzyme which is again going to chew off your things, right? So this, once you add this reagent to this reaction, this is your antibody which is being attached to your transcription factor. This MNase is slightly different. It's going to chew off everything that again does not remain. And at this step, right, at the final step, you can recover a lot more DNA as compared to what you had started with, what you would have gotten if you started with ChIP-seq. So the throughput is coming because there is a technological innovation that is happening at this end based on the enzyme that you're leveraging now to do the cutting operation. So if that particular endonuclease does the cutting operation, you end up losing a lot more cells or a lot more signal. Whereas if this does it, then you have a lot more information that is being sort of retained. So that's the key step. There is of course more technicality here which we don't have to get into. Okay. Any questions about this technology? ChIP-seq or cut and run allows you to profile in the genome where transcription factors or where are these acetylation or where are these histone marks being deposited. The readout for this signal would look like what you saw in this plot, right? So in this plot, what you're seeing, for example, is this is high because you got a lot of, when you ran the technology, you got a lot of reads, and when you went back and mapped it to the DNA, it all mapped to this location. So I know that this location has a K27 acetylation mark deposited. Okay. Sorry? DNA ME is DNA methylation. So it's down because usually at our 80% of our sequences already have some methylation mark deposited. If you want to be in the active region, then you want to remove the methylation mark. So that's what this shows, right? So that's active. Similarly, this is poly, so this is also towards the active region. Similarly, wherever you see the active region, there's a dip in the overall methylation levels. Okay. Okay. We'll skip this slide because that is just, I'm going to also delete this because this is highly technical. But just to give you one final preview into technology, if you want to figure out, for example, the technology that we have seen so far only tell you about where transcription factor is being bound or what are the acetylation modes. A very generic question that you can ask is, let me not worry about whether transcription factor is being bound or not. Let me try to study where my gene is in that vicinity, is the chromatin accessible? Can I actually access, is my chromatin accessible to other proteins? Like, can something come and bind? At this point, I'm not doing the binding experiment. I'm just asking a slightly more meta-level question that, is it even open in some sense? And those kind of technologies to profile this open chromatin regions, right? So for example, what is an open chromatin region? This region, there is not much nucleosome bound, so this is nucleosome free and it's also open. These regions, wherever you're seeing not a lot of overall deposition of nucleosomes, those are things where potentially transcription factors can come and bind. And you just want to understand whether your gene is in this region or in the closed region overall. So the way this is done, one of the ideas there, if you want to profile regions like this, is again, this is very busy slide. This is only for if you are really interested. I again would just want you to get the gist of the idea. So what is the goal? The goal is for me to be able to tell whether am I in a region which is accessible to chromatin, which is open chromatin. So if something is open, right? If you're in this region, these are the kind of things that you're interested in. This region or this region where potentially a transcription factor can come and bind. I'm not interested in which transcription factors are going to come and bind at this step. I'm just asking a very simple question that which parts of my genome, if I just take my DNA, which parts in my, let's say, liver are open and which parts in my brain are open. So to do this, you're interested in getting this particular region. You want to know the coordinates of this region and this region. How do you attack this problem is very, it's again, this is just a 10-year-old technology now, but extremely innovative. So the first thing that you have access to is a sequencing technology. Right? You can sequence anything that is given to you. Now you have to be smart about it. You have to be smart to say that I will only sequence things which are coming from regions that are accessible or that are open to the that are part of the open chromatin region. So while if I threw my entire DNA to a sequencer, it will sequence this entire thing. Now I'm saying that I'm going to only open sequence these regions or regions like this, which are part of open or nucleosome free or open chromatin. The way this is achieved is you very smartly is to sequence anything you require adapters, right? Like at some point you will put up your Illumina adapters because that's where the sequencing will begin. So this group engineered an enzyme which is called transposase, which actually does a cutting operation in the genome. It can cut DNA at specific locations and it can paste it somewhere. They engineered these TN5, which is called TN5. It's an enzyme. This enzyme was re-engineered such that it can get attached to Illumina adapters. Right? So this red and blue that you're seeing are Illumina adapters stuck to this enzyme. So now it can go randomly in any part of the genome. It will only be able to go to those parts of the genome which are accessible. Right? So for example, wherever you're seeing this axis, it can only access regions which are accessible to the chromatin. It goes there and it deposits an adapter sequence there. It will make a cut, and with that cut, so this is the cut that it is making. This is open chromatin. It comes here, it makes this cut. Once it makes this cut, it deposits these Illumina adapters. Okay, you can do there are some sort of overall gaps that are there which can be filled. Once those gaps are filled, you can just throw this off on the sequencer. And you would know which parts of your genome was this transposase able to access, because those are the regions which are actually also accessible to the transcription factors. Those are open chromatin peaks or open chromatin regions. Okay. Sure. So the goal clear? The goal of sequencing clear? We want to sequence regions which are, for example, we want to sequence very loosely. We want to sequence regions which are not being occupied by nucleosomes. Okay? Which means these long strings that you have, if you have these very beads on a string kind of stuff, and if you have a long string where there are no beads, you are interested in figuring out where are these. Yeah. Where are these empty spaces overall? Okay. So if I sequence this as it is, I will get everything which I don't want. I will get my full DNA sequence. I don't want that. I only want this. Okay? I only want regions which are open because there is nothing here.
But why do you want open sequences?
Why do I want open sequences? Because I know that if this is open, it's quite likely that some transcription factor would come and bind here. Right? So if I do this across different, so if I have, for example, two tissues which are expressing two different different set of genes. I want to understand why is it happening. Is it happening because the genes which are not being expressed happen to be in regions which are closed chromatin, or is it happening because they are indeed in open chromatin, but maybe there's a DNA methylation mark, right? Right? What is the mechanism by which certain genes are not being expressed? Okay, does that make sense? So to achieve this, you do a very simple operation, right? You bring in scissors. These scissors are interesting. I'll just draw the scissor like this. They come and they can cut this part. Very, very simple. But when they cut, they also have these Illumina adapters attached to it. They will just come and deposit them here. You just throw this off on the sequencer and you can sequence everything which is open. Okay. It is, I always forget what is attack. Accessible transposase. One second, let me recall. So this group has a this group always likes some war kind of mnemonics. So it's a forced mnemonic in some sense. I think it's accessible transposase of active chromatin. Now I'm curious because I've forgotten. See, everywhere they use ATAC-seq. So let me go to Wikipedia. Assay for transposase accessible chromatin. Yeah, assay for accessible transposase. They have something called spear-seq. Right, so they have also these kind of technologies which are which are named after these kind of attacking tools for whatever reason. Actually, it's pronounced as ATAC-seq. I call it ATAC-seq, but I think the right pronunciation is ATAC-seq. Assay for assay for accessible, transposable, accessible chromatin. So assay for transposable, accessible chromatin. Assay for that makes sense actually. So transposable, accessible chromatin. So this is transposase. It is accessing the chromatin. Makes sense. Yeah. No. These are Illumina adapters only. It's just that when they actually sort of thought about this could be any adapters for that reason. Right? The reason they are making this is it gives you a way of hold on, because when you actually do the sequencing, right? At this point, you'll still have fragments remaining otherwise, because these are random contamination that will come. This is the identifier which allows you to actually figure out what is it that you should do the pull down and you should sequence. So that is where the naming comes from. You cannot use Illumina, usually will not use it because it's a commercial. Right? So that's what the attaching fragment is. It's nothing but those adapters attached to it. Those are Illumina adapters. How will you map? Right. So at this point, this could have come from somewhere in the DNA, right? So if I take any fragment, we have done DNA mapping. So you use a similar approach, that you take these fragments, you will map it back to the DNA, and will map to unique locations where, and that unique location will tell you whether that is part of open chromatin region or closed chromatin region. You're only getting fragments from open regions, right? So if it maps, it's open chromatin. If there is no read coverage, that's closed chromatin. That's open chromatin, yeah, right? That's open chromatin. Okay.
So why this
Yeah. So in principle, it is. If you think about it, it is easy. Usually people don't do it. They usually will do it separately, right? And I don't know why. Is it because the reagents are not amenable, or it's not required? But I have not seen people do it within the same sample. You'll take 2 vials of it and do a separate experiment. Maybe because of the temperature conditions or the other stuff that is required. Maybe they are not amenable within the same sample. But you can always do it separately. As long as your sample came from the same vial, and you're making 2 different vials from the same sample, you will get concordant results, right? Because assuming everything is homogeneous, One sample will tell you where is the transcription factor bound. One sample will tell you where the chromatin is open. Okay. And usually where it is being bound it's a subset of the open chromatin regions, right? Because transcription factor can only bind in open chromatin regions. Okay, so that also brings me to this last part of the slide, which will soon be ending, is they can actually you can actually use this data to figure out whether transcription factors are binding or not. How do you do this? So that's also a very innovative approach, and actually that was developed in the same paper or a follow-up paper, but a very, very clever, mathematical and computational technique. So let's say this is my chromatin, and I'll just call it closed and open and closed. What I did not tell you is if you have indeed a transcription factor bound here, actually, right? Then when you actually make the cuts, so this is open, but this is the TF that is being bound there. Now, if this is open, if you think about the reads, then you will see, let me try to help you visualize it. So this is open, so you get reads like this, right? You get reads like this. Any ideas what will happen in the middle? This is open, this is closed, this is C, this is open. You'll get a lot of reads from here, right? So my question is that if I plot, let's say, the density of this, it will look like this. A lot of reads here, nothing here. And similarly here also there will be a lot of reads and nothing here. What will this look like where there is actually a transcription factor? What do you think would happen here? Why is this cut happening? Because you have a scissor. That scissor can access regions which are open to sides. Right? This is accessing all these regions. So the reason you're getting these reads is because there are so many copies that you have of this DNA. You made multiple copies. You add to it those scissors. So sometimes it cuts here, sometimes it cuts here, sometimes it cuts here, and so on. And that's why you get these kind of fragmented sequences. What will happen here if a transcription factor is bound? I'm just trying you to imagine, can you cut it? So let's say this TF, this is the TF, right? Let's say it occupies 12 nucleotides. Okay, and let's say, this is 15, and this is also 15. So if the scissor comes, it can come and cut maybe this. Sometimes it will come here it will cut this. Maybe it will come very close, they make a very small fragment. All sorts of possibilities exist, right? Again, your probability question. Given a length of 15 sequence, what is the distribution of the fragment lengths? That is where the question came from, by the way. The end-send question, the mid-send question, this is one of those questions. What is the average distribution of these fragment lengths? But what will happen here? Will you see a cut? Can a fragment come like this? It can, because I can make the cut here and I can make the cut here, right? Here. So you get a fragment like this. It will come very, very close and make the cut, and I'll get a fragment like this. But what you will notice is the amount of possibilities that exist here. Can I make a cut here in between? I cannot, right? Because it's not accessible actually. The transposase is blocked here. I cannot make a cut here. So here, overall, the fragments that start in this region are depleted. If I look at the distribution of the start sites where the cut happens, it will be very high here, it will be very high here. But this will be depleted because it's not accessible. So if it is not accessible, then you will see, and if the TF is bound, you will actually see a dip. If you just plot that, where does my fragment start? Okay, if you just plot the distribution of your fragment, this is a start, this is a start, this is a start. But here you will not see a lot of starts. You will see a lot of fragments still because, for example, this fragment can start here and here, but you will not see any fragment which starts from here, or from here, or from here, or from here. Right? So all this 12 nucleotide region where there is actually bonding happening, you will see a dip which would look like this, a valley which would look like this, if you were to plot the distribution of your start sites. Yeah, because how you're getting reads which can extend it, because the digestion mechanism when it actually cuts, right? It's random. The starting location where it's going to cut, for it, this location it can choose randomly. Okay, so it has made a cut here. So you have, so this fragment has gone away. You can sequence it. This fraction remains, but the second transposase can come and make a cut here because this is accessible. So you will get a fragment which starts from here and spans the TF. You can get it because this is accessible, this is accessible.
So you won't have reads which end in between the last.
You will not have, exactly, you will not have reads that either start or end here. Okay. So I just looked at the start location. You can make the same argument if you were to looking at the end location. Say that again. It's a very similar response, right? So that probability problem might appear as if it came out of nowhere, but these are the questions where you actually, once you look at the data, you have to model it. That what is the probability of seeing something that you're seeing, and that is at that point that's a computational problem, right? The biology has happened. You still have to connect this to biology. That why would you see something like that you're seeing, but once you see it, it becomes a computational problem. Okay, this will not happen in the end, this end sem, but that's because you've already shown you the reverse Bollywood approach, so you've already seen the solution to the problem. But this is the context in which it happens. Okay, so we are actually short of time, and I also have to do a quick review. Yeah, so this is just to show you that what is the difference between DNA-seq and there's something called assay. This I'm going to delete because I will not have time to cover it. This also we'll delete because we don't have time to cover it. But the goal is to tell you that overall, if you have access to these technologies, right? What the question that Devansh asked, that can you do it together? That also is happening soon. It's happening now. You can actually sequence multiple of these things within the same tissue or within now in the same cell, right? So the single cell version of ATAC-seq is called single cell ATAC-seq, SC ATAC-seq. You can do SC ATAC-seq, which tells you the chromatin accessibility within the same cell, and you can also do RNA, SC RNA-seq, or split seq or text genomics within the same cell, right? So this is happening at the nucleus level, right? Because your DNA is in the nucleus. You can use the same cell to do both the reactions. You can also measure the RNA, and you can also measure the chromatin accessibility within the same cell. This is something which we were not able to cover in the class, but if those concepts that we have figured out that we have covered in the class, if those are clear, this part is slightly difficult but not very difficult to analyze the data. Right? So I just wanted you to see the possibilities that exist. Any questions before we do a 12-minute review of whatever we have done so far in the class? Any questions about this part? So very quickly, today we saw methylation and acetylation and histone modification matters. We don't have an ideal technology which can directly go and measure it. We use a proxy. The proxy that we use is we target those marks by using certain other external quantities like reagents or antibodies. Once those antibodies come, the principle on which they work, you just remember the principle. The principle on which they work is they're able to sort of go and attack those locations, and then you bring in your sequencing and you sequence it. That's the sequencing, that's how ChIP-seq works, that's how cut and run or cut and tag works. Then came the idea of DNA methylation. DNA methylation we saw, again, we cannot measure it. So we again convert it to something that we can measure. So C gets converted to T. If it is not methylated, we sequence it. Then comes the idea of looking at just chromatin, overall open chromatin. Again, in an ideal world, we would just sequence it, which is what we are doing, but we are doing it very smartly by doing the cutting operation first, so that we only get regions which we actually care about, right? That clear? So you just have to remember these concepts, right? Because if you know these concepts about how to think about it, then if I tell you to design a new experiment where you want to profile maybe both of these things together, how do you go about it? Right? What is that principle that you're going to use? Principle you have to remember. Details I don't care about, right? Like I don't care about whether it's H3. Will you do chip seek versus cut and run? How much salt will you need? That is not to be worried about. But if there is something where you can do methodological innovation, how will you think about doing it? For example, GWAS for people who are missing the classes or anything of that sort. That's what I'm looking for. Okay. If I ask you to design, for example, that somebody figured out that a transcription factor. For example, if you figure out that this transcription factor, whatever, some TFX gives you advantage in crop resilience, right? So if this level is high of this transcription factor, then your crop will be resilient to heat. Actually, let's say, okay. So it's agnostic. It can grow in very extreme temperatures. How would you go about testing this hypothesis? Right? How will you design an experiment which will allow you to test this kind of hypothesis? What technology will you use? What experiment will you do? Those are the kind of questions that I'm looking for. Okay. Okay. We'll go actually back to the start. I don't have to talk about this. All you know, DNA is double helix, A, T, C, G. A double bonds to T, G triple bonds to C. You should know what that genomics is because you've taken a course in genomics. It's nothing but the study of genome. Genome is defined as everything which happens to be inside the cell overall. But different people have different definition. The distinction between genetics and genomics. Geneticists will study only one gene. A genomicist will study everything that exists inside the cell. Okay, we talked about p-values, we talked about probability, right? This is just a copy-pasted version of your slides. We thought about certain problems like that given a particular distribution, how do you think about coming up with a hypothesis and how do you actually test that hypothesis? This is usually done in this framework of p-values. We saw that the genome content can be different for different organisms. How do you computationally study something? We did exercises to figure out can we use just a very simple experiment to differentiate different organisms from each other? Is there GC content, for example, sufficient for us to tell that this organism came from one species versus the other species? You have done this particular example as part of your first assignment. You've looked at just again one metric, right? You're only looking at one metric, which is GC. And as we went along the course, we made this metric very, very complicated, right? So just GC was often not sufficient. So we innovated the idea of doing DNA sequencing so that we can sequence the entire thing rather than just looking at one summary statistic, right? So that's where the genesis of DNA sequencing happened, and the problem that this is it's a mix of we switched gears from just talking about biology to actually talking about dynamic algorithms or dynamic programming, doing some CS and some math and some statistics, looking at how sequences are conserved, trying to align two different sequences. We came up with a greedy approach or a dynamic approach to do this alignment by defining a score function between two different sequences and choosing the optimal path that leads to the best possible score. Okay. This was global alignment where we are trying to do it end to end. We also studied that in often cases this is not what you require, given particularly small protein sequences, or if for example, if that divergence has already happened. There's no point aligning things end to end, but probably better is to look for a region in the genome where maximum matching happens, and that's the problem of local alignment, which also we saw. Okay, I'm going to go over it very quickly, but if you have any questions anywhere, please feel free to stop me. You saw versions of Blast. You ran your Blast on your own name. You saw that it's a heuristic designed to speed up programming, dynamic programming, where it tells you that rather than if you have multiple such organisms or multiple such sequences, rather than wasting time doing alignment one step at a time, you figure out at what point should you start to align, right? So where do you figure out the seed sequences to do these alignments? That's what the innovative part of Blast is. A heuristic approach to figure out where should I even care about doing the alignment rather than doing this blindly. Okay, this is all of this is nothing new. This is literally what I did is I went through the lectures 1 to whatever 24, 23, and I picked out slides just to highlight what we have. So none of this is new and in such sense. We looked at the PCR, which you saw one or the other question come up in your assignment. There's one thing I think besides the bio people is something which hopefully will stay with all of you. It's one of those very, again, think about it, nothing very hard to think about, but very, very innovative idea which allows us to do whatever we have done in the class, which is to make more copies before you actually throw this off on a sequencer. Okay, so this is again copy pasted from the slides. You have different versions that we saw of different technologies, starting from standard sequencing to some amount of microarrays to second generation and third generation. We focus a lot when we actually align the data on second generation sequencing. The third generation sequencing, we did not download data and we have processed it, but again, the fundamental principles are not extremely different. The only difference that happens is because this was shorter reads, like 200 to 500 nucleotides, whereas these are longer, 1 kb to 8 kb long. The alignment problem is slightly different, right? Like if you think about, you prefer longer alignments now, and those need to be fast enough. But in essence, besides those that part, like nothing else is fundamentally different, right? So the reason I'm highlighting this point is to tell you that you should not think that this was not taught in the course. What was taught in the course was the fundamentals which will allow you to go out and apply these algorithms. Because any one course is not going to teach you every aspect of everything. But if you have those fundamental principles, you should be able to think that this principle will remain the same, right? Just the problem will change. And I'm saying this just because this happens in my life also, right? It's not that despite wherever I am, I still get to learn new things every day, and doesn't mean that I have to take a course to learn that thing. If you know the principles, you can actually figure out what should happen, bare minimum, right? So don't wait for somebody to teach you, teach yourself. Well, if you find somebody really good to teach you, you should, but don't wait for somebody to come and teach you things that you want to do. We looked at sequence by synthesis. We saw how these things are being, why it revolutionized the thing. Works on a very similar concept as standard sequencing. You allow reagents to be added. You allow fluorophore sequences to be added. The reaction happens, literally proceeds one base at a time, emits a signal. The signal is very low, so to amplify that signal, you have a chip. That chip has sequences that have been amplified. That's where bridge amplification is required, so that when the light is emitted, there is enough of it for the camera to capture. That becomes your bridge. That is the importance of bridge amplification. At each point, any such tag will emit a particular sequence, which will get recorded by the camera, and that's what the readout of your signal would look like. You keep track of this because at some point you're also sequencing certain fixed fragments. These are called your I7 and I5 indexes, which gives you the identity of which spot that molecule came from, so that you know that these molecules were all coming from one spot, so they should be used for determining the same base within a particular read. This gives us read, but people started to think about that these reads are very short and the process is not as efficient as it can be. Why should we waste time for the next base to come in and then remove the fluorophore and then again start the reaction? why not do it in one go? That's where the genesis of the third generation sequencing happened. You have one particular polymerase sitting, you have a very ultra smart camera sitting in a sitting in a zero mode waveguide. That zero mode waveguide allows you to sequence, to look at very small emittance of light that happens because the place where the camera is sitting is very small, so it can detect high intensity things, and it's one entire length of sequence that can be sequenced at once. In principle, the reason you want to prefer this is if you have in your DNA, if you're interested in understanding whether there are large scale deletions or not. There's nothing that Illumina cannot capture in principle, but one area where it does not do that well is if you have very large sequences that got added or deleted. Short read sequencing will not tell you because your aligner will get confused. But with this you can get read sequencing up to 8 kilobases, which is pretty long for the aligner to align. It should be able to align it confidently. So it's used a lot in these contexts where you want to figure out where are large-scale deletions or insertions. We also, of course, looked at nanopore. Nanopore is an extremely smart innovation. This happens because you literally have your DNA being passed through a flow channel. That flow channel emits current. Current is measured. That measurement can be converted back to DNA sequence. Then we talked about genome sequencing, how it is done. We had the Human Genome Project, which gave us a reference. All of our DNA is now aligned to that reference sequence. We shear the DNA before doing the alignment because none of our technology can full-on sequence the entire DNA, so we have to shear it. Once we shear it, we try to figure out again where does it belong by using the alignment approaches that we talked about. That alignment, however, the local alignment or the whole alignment is not an optimized problem for such setting because you will waste a lot of time doing O(n) square calculations with so many fragments. It will not scale. We thought about a new data structure which will allow us to do this very quickly, and that data structure used to be we saw multiple versions of this, but this is a combination of a trees and a tries and suffixes and Burrows-Wheeler transform. Central idea there being that you have a reference genome sequence, you already know what is there in that sequences. You take new sequences, which are your reads, and you try to just very quickly figure out, should I even care about aligning or not? Or should I just throw away all these sequences? That became possible because we were using this idea of suffixes. The suffix trees allow you to figure out whether should you even waste your time to do the alignment, because these are suffixes. If you don't do the first patch, then you're off. Then you just throw your sequences off overall. We saw this very cool innovation, which is called Burrows-Wheeler transform, which again has been sitting on our laptops for a while. This is what is used in the compression algorithms, lot of compression algorithms. The idea behind it is very unique, very simple. You start with suffixes, you sort them lexicographically. Take the last column and that becomes your PWT. That will allow you not only to do alignment because it has compressed the entire structure, but also allows you to get your own full sequence back that you want to do. Okay, then came time for going from or graduating from DNA to RNA. So we looked at the importance of why we study what we study. And I did cheat because I told you that the mRNA is more important, but of course there is an entire huge part here which I did not talk about, which is 96% of stuff, which is your tRNA and your rRNAs. And maybe you can take a different course, or maybe sometimes if we meet in the future, we'll talk about what is the importance of that part, but that's very important as well. But for this course, we focused on mRNA, which we captured using RNA-seq. Simple principle that RNA-seq that hopefully all of you will remember is RNA is in mammalian cells is a special quantity because it has poly-A tails. Poly-A tails can be attacked to sort of remove them from rest of the RNA that we saw. 96% which we don't care about for this class. We can, if you do that pull down, we can again do an RT step, reverse transcribe, DNA is more stable, so and that's what most sequencing technologies use. Convert this to cDNA. cDNA sits on the sequencer. You sequence it. So I'm going very, very fast, but please stop me if there are any lingering questions or any questions that comes to your mind right now. We thought about new ways to align this sequence because the previous approaches would not have worked, primarily because this problem comes because of now your reads being split because you have exons and no longer continuous sequences like what you had in the case of DNA. So we thought about novel ways of aligning them by looking at approaches like Callisto or STAR, for example, which uses the reference itself, where we have previously profiled the transcriptome to come up with a better index, which we use for our final alignment. We also use this opportunity to learn about a new algorithm called the expectation maximization algorithm, which allows you to figure out from the transcript level how do you go from one where you have absolutely no idea which isoforms are higher as compared to the other one, or which particular isoforms are more abundant. It allows you to resolve that just learning from the data itself, no supervised learning involved overall. We learned about once you get this data, how do you actually analyze it. This is a new plot which you would not have seen. This is just to show you different ways of doing dimensionality reduction. We only talked about PCA and SPD. That is not the end of the world. The exploratory and the dimensionality reduction is again a course in itself. One and two other different approaches there is something called an independent component analysis and a non-negative matrix factorization, which I did not talk about, but I'll encourage you to look up later on because that will be something that you can use in different settings. What we however learned in the course is how to think about dimensionality reduction. What are the principles which allow us to do it? Okay, and that principle was this recipe principle of PCA, but that's a recipe, but overall what we're trying to leverage is what directions have highest variance, which I can capture in my data. That's what PCA is. We then talked about very briefly that once you're given this data, you've done all sorts of exploratory analysis. How do you think about modeling this? Because this is counts data, so it requires specific kind of models to be able to differentiate between cases and controls. And that's where our paradigm of statistical testing came about to be, where we start to think about if I'm given RNA sequencing data from two different individuals or two different cohorts, how do I ensure one of them is controlled and the other one of them, or how do I ensure which genes are expressed differently between the two? That came from using this regime of p-values, doing different kind of statistical tests. Whatever happens, hopefully this table, this structure will remain in your head. You first question that you have to ask about your data, is it paired, is it unpaired, and then once you figure that out, you check for normality assumption. Does your data follow normal assumptions? If it does, you apply parametric tests like your t-test, which follows a parametric distribution. If it does not, you apply a non-parametric test, which is, for example, Wilcoxon or Handwriting. Again, we didn't see all versions of these tests, but the framework should be something that you should be able to capture. We don't have to, again, apply all the tests because then it becomes a statistics class and not a genomics class. We saw what is a p-value. We saw what are type 1 and type 2 errors. What interestingly we saw also is this multiple hypothesis testing regime, and I owe one explanation to you, which I'm going to post after the class, which is the error correction that we have done for multiple hypothesis testing. How does one of them work, which was very unintuitive, which is the FDR regime, right? So the multiple hypothesis testing here happens because we are testing multiple such hypotheses at once. We define something called family-wise error rate. This is one way to control the number of false positives that you identify. You correct this using Bonferroni. Very simple idea, just divide by the total number of tests you're doing. You control the total number of false positives that you will identify. However, we found that that's not always what you're looking for. You're looking for among the false positives, among all the total positives that I identified, how many of them are false positives. So it's not so much FWER, which is family-wise error rate, that you worry about, but you worry about the number of false discoveries, because we are interested in identifying genes that we can go and test in the market or come up with a test to deploy this. And that is better captured by this metric called FDR, which we corrected using this unintuitive approach called Benjamin-Hodgberg. And when I explained this in the class, I intuitively drew the diagram correct, but the explanation was not correct. So once you see the explanation put out in the vignette today, which is a computation exercise, I think that will become clear. Okay, so I will skip this for that reason. We also see Q values. I don't have an explanatory part about this, but this is another way to control the FDR. Most people will use Benjamin-Hodgberg. However, Q values have slightly more power overall to detect, to control FDR as compared to Benjamin-Hodgberg. Then came time to do single cell. We did, this figure is something that you'll be seeing for the first time, but it's a representation of what single cell is about. What your key takeaway should be is the identity of individual cell types remains as such. We are able to take a matrix of this very sparse matrix, 4% to 5% non-sparse values, and squeeze all information that is possible from it by leveraging techniques of normalization, where we can remove the technical noise that we see in this data. And the power of this technology is it gives you resolution to look at what is happening at individual cells. Right, so we saw that if you did this kind of experiment, you will not have covariation structure. You see that in single cell. This is also what we saw. We saw exponential, or beating Moore's law in terms of the number of data sets that is growing. And there's a particular reason I'm highlighting this plot, and I will come to that very, very quickly. We saw different models to normalize this data. Y Poisson is often not the optimal model, which is theta equal to infinity. And we often resort to using negative binomial to model this kind of count distribution. Finally, with Shweta, we talked about how to genotype individual. This is a new slide, which you might not have seen, which captures the essence that she talked about. You have people, 10,000, 1 million, which have different genotypes, which means their DNA sequences are different. They also have different covariants or comorbidities. Somebody has diabetes, somebody has maybe some liver disease, some kidney disease, and so on. We are trying to draw association that if I see a particular variant come up again and again, is it associated with diabetes or is it associated with X disease? That's what your GWAS is trying to do, genome-wide association studies. We are taking the full-on genome sequences using arrays and trying to figure out that if I have the genotype of 10,000 people, are all of them A, and do all of them have type 1 diabetes, which means that having A at a particular location is associated with diabetes. We did not assign causality to it. And today we talked about some models for deciphering gene regulation, not statistical models, but overall, how do you think about doing gene regulation study in all of this context? We talked about epigenomics, which we finished today, so I'll not go too much into it. Quickly slide through it, right? But there is a reason I showed you some of the slides, but where do you go from here, right? So if this is the first course that you took in genomics, where do you go from here, right? Some of you who are going to graduate, of course, what is it? What is there in this field, right? The reason to do this is because it's just the way we are taught biology in this country is it's still not, it's a problem hopefully that will get solved in the next 15 to 20 years. But hopefully you'll appreciate that they're not two orthogonal lines. Math and biology are not separate. They are actually very intertwined into each other. And if you go back and look at lecture 1 slide, you will see that one of the problems that we started was the codon problem, where from 64 codons you get 20 amino acids. How does that happen? That's a mathematical problem. The solution came from a different approach, of course, but it can be thought of as a mathematical problem. And I want you to keep that account, always live in your head, that these are not two separate fields. One can feed into the other. So there are a few courses that selfishly I would recommend to take, which is, so I'm not going to run actually next semester, but there's a course called Computational Genomics of Aging, where we use principles that we learn in this course. It's right now open to everyone. Anybody who has not taken this course can also take it. And we try to do the rich people's science, which is we try to understand why aging happens, what are markers, hallmarks of aging, and is there any science in trying to think about, can we actually reverse it? Is it even reversible? Is that a legible question or not? There's a course which I've never run, which is Introduction to Data Science for Genomics, where all the methods that we touched in this class, which we actually did not, go into depth into, is what that course focuses on. I've never run it because I've not had enough bandwidth to be able to support it. But if I'm running it, of course, and if you're still around, then you should. I would recommend that you take it. But again, we live in very interesting times. You don't need me or anyone else again to teach you. There is a lot of stuff out on the open. There are courses on Coursera. There are courses, so many courses now on YouTube, whatever you want to look up, you should be able to find, right? So that's from a learning perspective, that if you want to go deeper, in fact, there are much better educators out there on YouTube if you go and search. Okay, these will be your seed courses to help you figure out. The second one is a very interesting course which I highly recommend. This is from people. This is from the Moderna people. They talk about how mRNAs, how that entire vaccine development process goes on in the mRNA world. That, by the way, was a computational problem, right? Once you figure out the sequence, it becomes a computational problem. Okay, if you're ever thinking about, or if some of you are thinking about either grad schools or thinking about the next approach, this idea that I came from engineering, so I cannot do science, or if I came from science, I cannot do engineering, no longer holds. If you're going to a good school, they will admit you irrespective of wherever you come from, unless there is extreme competition and they cannot, right? So I don't think anyone in this age is foolish enough to reject somebody just because of the training, just because they did not take X course in biology or X course in mathematics. That no longer holds, because we can all there is enough resource out there to train yourself in the process. This is especially true for PhD because it's a longer journey. For masters, I don't know, but for PhD, usually you should not have trouble getting into any of these departments and still be able to do biology. So this is not an exhaustive list. There are people in electrical engineering who do genomics. There are people in computer science, of course, physics as well, chemistry, genetics, statistics, applied mathematics. All of these people actually work on genomics problem. This is partly true for our campus also, not as much, but if you go out, you'll see people doing genomics in different departments. The other advice I have for you, especially for people who are looking for grad schools, while ranking is good, you should look for the ecosystem that your school provides. It allows you to flourish. If you don't like the frequency, just don't go there. It's very simple, because you have to spend there 5+ years, or at least 5 years, and you want to be in an ecosystem where you feel happy. If you're not happy, you'll not be able to do good research. That's the bottom line of this. This is not so true for masters. Masters is quick, 2 years you're done, then you move out. But particularly for PhD, right? That's very important. This is just for people who are planning to go for PhD. This is for picking up the right skills. No program will train you in everything. PhD is to go about depth, but PhD is more about getting the skills right? So spend enough time, the kind of time especially that you get in where you are right now, sitting on the other side. I can tell you from experience, you will never ever get that in life. And that's, so have fun doing whatever you choose to do. You don't have to go to grad school. You can go to industry, for example. With these skills, there are a lot of companies actually right now in India particularly looking for people who train in these kind of areas. Strand Genomics being one of them. I'm not paid by any of these companies. So Map My Genome, MedGenome, Precision Health, and again, this list is not exhaustive. These are just suggestive startups and companies. Strand Genomics now is owned by Reliance Healthcare, by the way. So startups or corporate companies where these skills will be useful. You can go to, if you're thinking about abroad, these are again some of the suggested companies, Genentech, 10x Genomics, Illumina, 33andMe, which I showed you, Quagen, Thermo Fisher is there in India also, but Mammoth thing, don't go to Thermo Fisher in India. Again, this will be recorded, so I will get attacked at some point. But Mammoth is an interesting startup trying to resurrect mammoths, right? So this is again a computational and a biological problem. Look up this person called George Church if you're interested. That's where this idea came from. Well, you can also think about your own startup in this space or a related space. There is a lot of room right now, particularly in India, to fill. Just to give you some ideas about what areas people need help with, is analysis as a service, for example, is a clear idea where, if you open a company which does analysis as a service and you do a good job at it, you'll get a lot of money without actually having to spend anything, because a lot of this is open source. You don't have to pay anybody to do anything, but you can make yourself money. The other area where you can actually do a lot of innovation is if you have a knack for doing some experiments, and I can give you advice, but I'm not the best person to give you advice, because I should have taken this advice and opened a startup, but I don't have a startup of my own. But I think there are areas which definitely require, if you want to solve, then you should be able to solve. One of them is some of these technologies are actually benchtop. We don't need to go to a commercial player to be able to do an experiment, which a lot of labs, for example, in India, or even in both industry, have to rely on. So if you can come up with very frugal ways to do some of these experiments that I talked about, if you recall, the principles are not very hard to understand. But why do I have to depend on a commercial kit to be able to do this? Why can it not be done in a very frugal fashion? Why do I have to wait for reagents coming from outside India to do a very simple experiment? If you can think of innovative ways where that problem can be solved, that's $1 million problem in the Indian ecosystem. Okay, so wherever you go and whatever you do, this is the one simple advice that I passively absorbed, and I just tell people, just show your work and just don't tell. So showing is, you did this course, and I think some of you already have GitHub repositories of this assignments on your GitHub, so which is good. You have something to show, but all that is open source. Talk about the stuff that you've done rather than just saying that I did this course, right? So doing something doesn't mean anything unless you can show that you actually know your stuff. So however you're approaching, try to sort of put that idea across. And now I have a final thing to admit, and I will end in 2 slides. I actually did not finish what I started. And the story, if you remember, started from being able to solve this sickle cell anemia disease, right? So I told you that a solution exists and FDA has approved it. But actually nowhere in the class, after looking at that first assignment or the first hands-on, did we ever come back and revisit this problem. This problem was a very simple problem where there is one single mutation which causes sickle cell anemia. And what I promised to you is that at the end of the course, I will be able to give you the solution to how this is solved. And because of just one lecture short, we were not able to do it. But I think I should finish the story and leave you with a bonus lecture which you should go back and read, which is at the end of this slide, which will actually finish the story. But let me give you again the preview of why this story is important and why was it complete. So this story has 2 components of the course that we talked about. One is GWAS, where people had studied, done these kind of association studies in sickle cell anemia patients along with normal patients. And what they had observed is there was another gene called BCL11A. If that was mutated, for example, like if it had a particular mutation at a particular site, then those people had that kind of genetic profile were less disposed to having a sickle cell anemia. Right? So if you follow these people overall, then you will see that these people who have a particular mutation, for example, in BCL11A gene, were less predisposed, or their progenies were less predisposed to sickle cell anemia. And they discovered that, okay, there's something definitely important about this gene, and what was studied then is this gene BCL11A is a silencer. If this gene is on, then it suppresses a particular kind of hemoglobin, which is called the fetal hemoglobin. So when you are born as a fetus, when you're a fetus, then there is a certain kind of hemoglobin that your body is producing. As you grow into a baby and an adult and so on, this gene, this fetal hemoglobin will be switched off, and there will be an adult hemoglobin which will be switched on, right? So this is done by this gene called BCL11A. The idea, however, then was that if I want to treat these sickle cell patients who are synthesizing a wrong version of the beta hemoglobin, but when they were growing up as a baby, it was all good because they had that fetal hemoglobin where there is no mutation. That is normal because they were able to develop normally, otherwise they would have been dead in the womb itself. The idea was that, okay, can we now go back? That I know that BCL11A is the gene which has this mutation which suppresses the fetal hemoglobin. Can I go and reverse this mutation, or can I go back and remove this suppression power of this BCL11A and switch it on back again? Repairing one particular mutation is hard, but switching on a gene is easy. So if you want to switch on a gene, this was a much easier problem to solve rather than going and correcting that location. Because if you switch on this gene, you're done forever, right? You don't have to worry about it ever all over again. That is a process that was followed in this therapy, which is the FDA approved therapy. It goes on and it switches on that gene, switches off the BCL11A gene, which in turn leads to production of your fetal hemoglobin, which makes up for the lack of good hemoglobin that sickle cell anemia patients are producing. So without actually doing an edit in place where you should have done an edit, you're able to treat people who have sickle cell anemia because you are making up for that wrong hemoglobin by switching off the wrong gene, switching off the gene which is shunting off your fetal hemoglobin. Right. So very interesting and a very innovative solution without actually hampering anything that is happening in that particular location to reverse this. Okay. So I will end here, and there is a full-on lecture on CRISPR which I unfortunately don't have time to talk about, which covers why that kind of process is important and how is that reversal happening. So stop here. If there are any questions, please let me know. Sorry I exceeded by half an hour. Any questions? Okay, great. Thank you and good luck.